Okay. I have cheater notes here because I must confess that our wonderful committee manager, Stan, made the slides, and they're very minimal, and I'm not a very minimalist slide person myself, so I have cheat sheets. Okay. So basically, as mentioned, this is a case study, and I'll be talking about how we came to look at this, why it's important to address it, and what is the project that we're doing to address the issues. So a little bit of history here. In 2006, the T-Gennaro molecule 1412 produced a massive cytokine release in 6 healthy human volunteers on a clinical trial. It's a CD28 superagonist, which means that it has the ability to stimulate T cells without requiring co-stimulation. None of the preclinical work, including the human in vitro assays that were conducted, were able to predict this massive cytokine release that happened on the clinical trial. And as a result, there was a whole lot of scrambling afterwards across the industry and academic institutions to develop and optimize cytokine release assay formats. Excuse me, most of these were independent efforts, but Richard Stebbings probably was the most prominent and published the most on these efforts. So this led to a workshop in 2009 conducted by the EMA, and the, excuse me, essentially the outcome of the workshop was 3 predominant conclusions. One, cytokine release assays, despite all of their challenges, do have a place in hazard identification, although perhaps not so much in risk assessment per se. They could not endorse a particular assay or assay format. And why is that? Because differences in mechanism of action, differences in the molecules themselves, and the targets require the ability to have flexible platforms. So the HESI Immunotox Technical Committee decided to take this on as a work stream, and it was multi-pronged, starting with a survey and a workshop. And I guess Madeline Fort has been the subcommittee leader from the inception. But anyway, they started with a survey of member companies, excuse me, and then conducted a workshop. And what did we learn? So we agreed with the EMA conclusions that there is no really gold standard assay that should be conducted. There are different formats, and they're all relevant depending on the molecule being investigated. The decision on what assay to conduct and how to conduct it depends on a number of factors, including the mechanism of action. Could be the target, the cell population that you're going after, any potential for cross-linking or not cross-linking. Is there an FC-related mechanism contributing to the potential? Are soluble factors involved? Interaction between endothelial cells, etc. And so basically, there's a need to tailor the assay for the particular program. Within a given approach, though, across the industry, there are different conditions. So there are different standards being used, different dilutions, various aspects. Do you do it during the full moon or not? A lot of it is what works in your particular organization. And companies use the data for different things. So some will use it for a go, no-go decision. They may use it to help determine what the pharmacologically active dose might be. Some will re-engineer the molecule to try and minimize it. There may be an influence on the first-in-human starting dose and how to escalate, or whether there's a need to escalate, all sorts of things. So this, the workshop then resulted in a publication. resulted in a publication which actually has just been accepted in Cytokine. Not sure when it'll be actually published, but literally last week Stan sent out the announcement to everybody that the publication was accepted. So as the next step, basically Sandrine Vassilier from the National Institute of Biologic Standards and Controls in the UK proposed a collaboration with the ITC to look at various standards. So why do we want to take this on? Basically, the opportunity to try and have some sort of qualification of platforms to develop more confidence in our data. Did I miss something? Okay. To develop some sort of future confidence or context. So we would be able to compare standards within a company and do they perform in the same way as these standards, but we'd also be able to develop an ability to compare with the data with historical information and develop some knowledge. And, you know, as much as it's not really possible to harmonize the assays, this gives us an opportunity to have some consistency across assay platform by having everybody use the same controls. So the institutions already have optimized in-house protocols, which is why we're not asking people to try and come up with a standard assay or change their current methodologies. But we do need to have a set of standards which can fit into the current assays that are already used. And we're also looking at a response range, so low, moderate, and high responses, and that will help us compare our own molecules to those types of responses and at least have some idea how severe a reaction might be. There will be a standard repository at these standards at National Institute of Biological Standards and Controls, which we'll all be able to use. So the basic assay is put drug in cells, in wells, and incubate and measure cytokines. But there's a variety of assay formats, and this is done with or without mobilizing drug and/or the cells. So what are the standards that we'll be using? Well, the number of the counting shall be 3, to quote Monty Python, and that's the theme of the day. Anyway, we're using 3 different positive controls, one producing mild cytokine release, 1 moderate, and 1 severe. And because 6 is the new 3, we also have 3 negative controls. And these are isotype controls for the positive controls. So as I said, we have 6 controls, and we have 10 participating companies. And I don't have a slide for that, but if you're interested, it's Amgen, Pfizer, Merck, Roche, Celgene, Novartis, Janssen, MedImmune, GSK, and Genentech. And so there are 10 companies on 2 continents. And basically, they'll all be measuring their own internal panel that they usually use, using their own methodology with the standards. But the one constant in terms of cytokines being measured is that all will measure IL-6, IL-2, tumor necrosis factor, and interferon gamma. So they'll then send supernatants from their assay, aliquots, to NIBSC, who will run their own assays with their own cytokine measurement methodology. The goal is to start gathering data in June, and I know several of the companies have already begun this effort. We hope to have all of the assay work done by September, end of September, and hope to publish in 2017. So basically, what is the hazard we're looking at? Cytokine storm. This effort should be pretty efficient. It's a fairly quick study, and we're incorporating it into an existing methods, so we're not reinventing the wheel. Developing this set of standards, and this will allow some degree of harmonization and greater confidence in the data. And because of the confidence across platforms, I guess that's how Stan fitted the integrity, the I, into HESI. And I do want to thank Stan and Oscar for supporting the committee, and Connie has as well over the years. And also definitely Sandrine, because she's put a lot of effort into this, as well as Madeline Fort and the rest of the people that are on the subcommittee. And there's a fish on this side, so I think I'm done. Any questions?
Thank you very much. Open for questions.
That's because I didn't show any data. Oh, John.
So, I assume the standards are going to be, it always seems like there's a lot of variability from all the standards and response. So if you guys have built like a huge pool of this that will be kind of like the benchmark for years to come.
Yeah, Sandrine promises that that's what they'll do, that they'll be able to make these available, and that is part of the mission of her organization.
There's a question here, Jim, first.
Yep, just thought I'd come to the microphone as told. So it's not really, it's a comment more than anything else. I mean, cytokine storms are a problem for cell therapy in terms of certainly managing them and as a major risk factor. So I think with the newly formed cell therapy tracking committee and the committee you've just been describing should certainly get our heads together to look to see what we can learn from one another, or certainly what we can learn from you guys in looking at these aspects.
The paper should make for some really good reading, because it discusses a lot of the inherent challenges between, the variability in the biology itself, the variability in the molecules. What do you do if your target is only on activated cells? Well, how do you do an assay to see whether you're activating cells if the target's not on there, and if you activate them to make the target express, then you may get cytokine release just because you did that. So it's a pretty challenging question.
So thank you very much. It was an excellent summary, and the work of this committee is one of the reasons that our company continues to re-up every year. Two questions. You mentioned lack of predictivity, and I'm wondering if you meant absence of signal completely or inability to predict the sensitivity of the species. And then related to that, if that's true, are there any examples with CD28 or any other molecule where you can replicate a difference in species sensitivity with an in vitro cytokine release assay?
So I was really careful to say lack of predictivity for massive cytokine storm, because there were hints in the preclinical data that something was up. And actually we have looked at least at Pfizer, and also some of the papers related to the CD28 superagonist, they did look at differential responses across species. So it has been done, and it can be done. First you have to find the right platform, because if one of the reasons that they didn't see a difference in the response in vitro monkey to human with the CD28 superagonist was that they weren't using the right platform. So once they did, they were able to see that the monkeys were less responsive. But nonetheless, one of the conclusions from a lot of that work was that monkeys aren't relevant for cytokine storm, and I'm going to take this platform opportunity to say that don't believe it. It depends on the target, so for this particular target, monkeys weren't relevant for predicting what would happen in humans. But, we've seen that it depends on the relevant, whether the target's expressed, whether it's on the right cells, and whether it has the same biologic effect in that species. But monkeys are quite capable of producing cytokine storm.
Thank you very much indeed, Ellen. Thank you.